Good evening, ladies, and this is my second podcast on DNA. So, today we'll be talking about the three types of bonds in DNA. So, we know in biochemistry there are non covalent forces and there are covalent bonds. The non covalent forces between the two strands of DNA are, of course, the hydrogen bonds. So, a hydrogen bond involves, of course, hydrogen and either a nitrogen or an oxygen in case of DNA. So, nitrogen and oxygen are highly electronegative. They pull electrons towards themselves, giving themselves a partial negative charge, whereas the hydrogen gets a partial positive charge. We call that a delta positive and a delta negative charge, therefore forming a very weak bond between the bases in DNA. A and T are held together with two hydrogen bonds, and G and C are held together with three. Another kind of bond is the covalent bond, which is very strong and it requires a lot of energy to break these bonds. So in DNA, as we know, in the backbone there are phosphodiester bonds connecting the phosphates and the sugars. Another covalent bond is the N-glycosidic bond, which is present between the sugar and the nitrogen of the base. So that's all for now. Just remember three kinds of bonds in DNA. We have hydrogen bonds, phosphodiester bonds, and N-glycosidic bonds.